All right, welcome everyone. This is part one of the lecture on whole body metabolic rate. So, I guess we want to start by situating ourselves again on organizational biological scale. So, we've come a long way in the first 4 weeks. We started off talking really at the atomic level, started moving up to the level of different biomolecules that Roja was talking about, membranes, organelles like mitochondria. We started moving all the way up to the level of the cell. And then if you take a bunch of cells and you put them together, you get tissues. Roja talked about things like muscle contraction, muscle tissue. And if you get a bunch of tissues together, you can have an organ. And if you have a bunch of organs together, you can have an organ system. And so we moved to that area. Roja was talking about the nervous system and the muscular system. And where we're starting to move now, this week, is the level of the individual, the level of the whole organism itself. And we'll talk today a little bit about metabolic rates at the level of the individual. Eventually, we'll start moving up into populations, communities, and ecosystems. But for today, where we're situated is at the level of an individual. So, this course is energy flow through biological systems. And so that's the currency that we've been talking about. Next week, we'll talk about photosynthesis and organisms that procure their energy from solar radiation. That second line there, chemosynthesis, we're not going to talk too much about that. Those are organisms that are getting their energy from chemical sources, things like hydrogen sulfide. These are organisms down, say, at the bottom of the ocean, hydrothermal vents where there's no sunlight, and so they've got an alternate source of energy. But what we've talked a lot about this term is cellular respiration. How organisms are taking chemical energy from glucose and turning it into ATP. And that covers all the animals, lots of the plants, most organisms that you know of are doing some form of cellular respiration. And in the last week, we started talking a little bit about the products of respiration, specifically ATP. What can you do with ATP? Right? And so Roja talked a little bit about action potentials and muscle contraction. I talked a little bit about heat and thermal biology. And so that's where we're going to talk today, more at the level of the whole organism. So there's the summary equation for cellular respiration. It's not key that you know all the details of the chemistry, but we want to know generally what goes into this summary equation and what comes out. What are the products and what are the inputs, right? So we know that glucose and oxygen are going in. We know that we're getting CO2, ATP, and heat out. That sort of thing. Can we measure those inputs and products at the macroscopic level? That's what we're going to talk a little bit about today. So I know Roja put a slide similar to this up, just showing all the detail that you can get into with the biochemistry of these physiological systems. And it's true. When we're looking at cellular respiration, there's just a whole lot of detail that is important, but we are not going to cover it in 126. But just keep in your mind that all this stuff is going on when organisms are doing respiration. It's going on inside of an organism, and that's, I think, what I want to stress here today. Don't lose sight that this is all happening within the cells of an organism. So if we've got a bald eagle here, yes, we have to realize that all of those things in that flowchart are going on. But how do we actually measure that in a natural system, especially in field situations where lots of these wild animals, it would be very useful if we could do that. Well, there are different ways you could approach it. You could just do something simple like measure the total amount of energy going in and the total amount of energy going out, right? You could look at the food, and you could look at the poop, and you could do some sort of calculation to see how much energy budget is changing for that bird. That's okay, but it's a difficult thing to do. Diet is not an easy thing to quantify. So knowing how much an organism is eating every single day is difficult, but probably more challenging is knowing the composition of that diet in great detail. So that's a really hard challenge for anything kind of outside the lab. Any organism in any sort of natural system, that's going to be a really difficult thing to quantify. But that's not even the hardest part. The energy out part is very difficult as well. So there we've got an eagle in the intertidal zone, and it is defecating, but that is liquid poop. So good luck trying to collect that and quantify that several times a day out in the field. So energy in, energy out. It can work in some lab situations, I think, where you're very controlled, but it's a really difficult one to use for most species. So let's look at that summary equation. What sort of things can we measure from that? Well, we could measure ATP produced, right? That we've talked a lot about ATP, and you could do that, and some people do that. But you're looking at the ATP produced by all the cells in that entire organism. That's a really difficult thing to try to monitor, especially in a living organism. So in most situations, that's not really a useful measure. You can measure how much heat is produced. We said that heat was a byproduct of cellular respiration. And you definitely could, organisms do shed off heat. So maybe you could take a thermal image or something of that bird. Problem is that bird is in a complex 3D shape, and it has depth, and so temperature is going to vary across that shape and then with depth from the inside out. So it's very difficult to make an accurate measure of heat at the level of trying to correlate it with cellular respiration. So that's, again, a really difficult one. Water is produced as well, right? We talked about the electron transport chain and water being kicked off at the end as the end part of that process. So yes, water is produced, but the amount of water produced by each cell is pretty small, and you're talking about an organism that is largely made up of water already. So being able to pick up the signal of water produced by cellular respiration is a pretty tough challenge because you're talking about a vanishingly small amount of water relative to what's already in the organism. So that one's not that useful either. So what typically we do is we focus on the gas exchange part of that equation. So oxygen usage or CO2 production. Those are two common ways that people measure cellular respiration in all sorts of organisms. And so people do those sort of gas exchange measurements to make estimates of metabolic rate. And one of the first terms I want you to know is basal metabolic rate. So that's a common way of measuring physiological activity. It has to do with taking gas exchange measurements from subjects that are calm and not stressed. So while they're at rest, while they're at a comfortable temperature, while they're unfed, so they're not going through digestion, and just generally not stressed. So here's an example of that. You've got a subject who they're measuring gas exchange through that tube, and the subject's reclined. They've cut off the light, and just in general are trying to take away all external stresses. So you're getting the very basal metabolic rate that that subject requires just to maintain its their biological systems. And people have done that for all sorts of species. And when you plot them out in a plot like this by size, so on the x-axis is body mass in kilograms, and on the y-axis is basal metabolic rate. So if you do that, they come out incredibly linear. So you end up with a line with a slope of 0.75, and that is a really robust result that occurs across many different taxa, across many scales of size. So everything from bacteria all up to whales. You get this massive sample of different sizes, and they all fall on this sort of 0.75. And even within a species, you still see that that 0.75 slope. So this is a really, really generalized result. But basal metabolic rate related to body mass. Now, this is one way of looking at physiological productivity, but another way is to do a graph like this. And so it's similar, but the y-axis is slightly different, and that makes all the difference in this graph. So maybe just take a second by yourself, pause the video, and just look at the units and see if you can make sense in your head before I go on what the difference between these two graphs are. Flip between this graph and the last graph. Okay, so the biggest difference is that this graph, the y-axis, the metabolic rate, is divided by mass of the body, kilograms. So it is the specific metabolic rate. It is a standardized measure. So in the first one, we just had basal metabolic rate, the straight metabolic rate. This one is standardized. And so what you see here is that for the smallest organism of the shrew, you have the highest specific metabolic rate. And the elephant actually has the lowest, which is completely opposite of what we saw here. So if you just look at like the total metabolic rate, the basal metabolic rate, elephants are just so big, they're going to have a higher metabolic rate. But if you standardize it by body size per kilogram, the shrew actually has a higher metabolic rate than the elephant. So these are two important measures, but they're slightly different. And it's important to know that the shrew is maintaining this very high metabolic rate relative to the elephant because they have different sizes and different life strategies. And so sometimes it's more useful to look at this standardized measure to make fair comparisons across organisms that are of different size. And one of the things that matters here is basically a surface area to volume argument. That surface area scales to the exponent 2, volume to the exponent 3. And so what you end up is, as you get larger organisms to the point of an elephant, it has a lot of volume, a lot of inside body mass, and not as much surface area. And this becomes a problem as it gets bigger and bigger because each cell is producing heat from cellular respiration, but it doesn't have enough surface area to lose that heat to the outside air. And so at some point, it has to have a very low metabolic rate so it doesn't overheat. If it had the same metabolic rate as a shrew, it would basically start on fire. It would be too hot. So the elephant can be really large in size, but it doesn't have enough surface area, so it has to have a low metabolic rate. And you can see that in elephants because elephants are trying to lose that heat all the time. It's why they have those big floppy ears. They're just trying to make extra surface area so that they can shut the blood in there and just lose as much heat as they can. So the other part of that is to run that hot, the elephant would have to eat an immense amount of food to support that physiological activity, to support that metabolic rate. And so they probably wouldn't be able to find enough food to support themselves each day. So there's important ecological consequences, physiological consequences of size, and they're related to volume, surface area, relationships, and metabolic rate. All right, so that's basal metabolic rate. Another common measure you may see is called maximum metabolic rate. So same idea, people do gas exchange studies, and in this case, instead of making the subject comfortable and not stressed, you run them out as hard as you can to see at what point do they reach their maximum metabolic rate. And so treadmills are a common way of doing that with athletes. Put the gas mask on them and just run them out to their maximum. We do similar things in aquatic respiration. So we have things like fish tunnels where they basically turn the flow up and run fish at their maximum swimming speed. And so you get something similar like that in the aquatic realm. So while they're not breathing like we are in the air, you can still measure changes in oxygen concentration within the water, like the dissolved oxygen concentrations. So, and those two measures are important because armed with just those two measures, you can plot a basal metabolic rate and maximum metabolic rate, and you can estimate the difference between the two lines, and that area between is known as something called the aerobic scope. So here in this graph, we've got temperature on the x-axis and oxygen consumption on the y. The red line is the maximum metabolic rate, and the green trace is the basal. You can see at very cool temperatures on the left side, there's not much basal metabolic rate, but the maximum is quite a bit higher. And so there's a lot of space in between those two lines where an organism could exist and comfortably respire. But as temperatures get much warmer, now the basal metabolic rate starts really increasing quickly, because even if you're not doing anything active, you're having to do things like sweat, right? We talked about that last day, to try to keep your body temperature down. And so you're having a much higher basal metabolic rate. And at the same time, the maximum metabolic rate is plateauing because there's only so high you can go. And at some point, they're going to meet. Those two lines are going to meet. And so as you get to warm temperatures, there's much less of an aerobic scope available for the organism. So there's not as much room for that organism to exist comfortably from a respiration point of view. All right. Okay. So I think I'm gonna stop there for video number 1. I'll post a small second video, but I think that gets us started on metabolic rates at the scale of the organism. All right, so stand by for video number 2.